Welcome to This Explainer. Today, we're embarking on a pretty epic, almost cinematic journey right into the microscopic world. We're going to uncover what I like to call the ultimate secret in Cambridge Biology 9700. Because, let's be honest, we really need to bridge that frustrating gap between simply knowing your biology facts and actually securing those top band marks on exam day. So think of this as your diagnostic masterclass. We're literally peeling back the curtain on exactly how examiners think, and I'm going to show you how to take that dry syllabus knowledge and transform it into structured, undeniable evidence on your exam paper.
Okay, let's dive into this because this right here is our central real-world mystery. How is it even possible that you could perfectly memorize every single page of the textbook, walk out of the exam room feeling super confident, only to get your results and see those marks somehow slipped away? Well, you've got the knowledge for sure, but the syllabus requires a highly connected conceptual understanding. The principal examiner, they aren't looking for a human encyclopedia, They are looking for analytical depth. So to solve this mystery, we have to stop just reading the material and start investigating it like a detective.
The solution to our whole mystery actually lies in understanding the traps. Coming straight from the principal examiner reports, here are the top 3 culprits for lost marks. First up, missing units in calculations. It's a tiny, heartbreaking omission, but it costs you really easy marks. Second is using incorrect terminology. You absolutely have to define your terms clearly before you use them. And third, and this is a big one, providing generic answers that aren't specific to the given context. To combat all of this, we're going to use something called the method mark strategy. It's basically an approach where you get awarded marks for your logical, step-by-step process, which proves to the examiner exactly how you arrived at your final conclusion.
Imagine looking through a cinematic microlens right now, just plunging deep into the nucleus to witness the DNA double helix. It's this hyper-realistic, dynamic structure that's bound together by hydrogen bonds and those anti-parallel strands. Now, to secure full marks when you're asked to analyze this magnificent structure, we use a high-mark framework. It breaks down into 4 really essential phases. Step 1 is concept. You have to identify the underlying biological principle. Step 2 is evidence. Extract specific values or trends straight from the data they give you. Step 3 is reasoning. Connect that evidence right back to the concept with clear causal relationships. And finally, step 4 is wording. Refine your answer using the specific keywords from the 9700 syllabus.
Let's actually apply this with a red pen versus green pen analysis. A flawed red pen answer often falls into the trap of cause and effect confusion. That's when you say things like, the plant makes energy, instead of explaining the precise causal link. It misses units, it uses vague language, it's a mess. But the green pen top band answer? That one uses specific terms like complementary base pairing. It cites precise data points, and it establishes completely correct causal links. Remember this golden rule. Vague answers are empty answers. You literally must link your theory directly to the biological consequences.
Now, what's really interesting about this is just how high the stakes get in topic 8.1, blood and immunity. This section accounts for nearly 15% of your paper 2 marks. When you're analyzing the structures of red blood cells, monocytes, and lymphocytes, you're very often going to face complex calculations or data interpretation. And here is where the method mark acts as your ultimate safety net. Examiners actually award marks for the correct systematic process, even if your final numerical calculation ends up being slightly off. So, show you're working. That's how you secure the foundation of your grade.
To really master comparative analysis, you need the right statistical toolbox. So let's outline your critical instruments here. If you are comparing the means of 2 continuous data sets, you pull out the t-test. Analyzing categorical data for goodness of fit? Well, that's going to be your chi-squared test. And if you need to determine the strength and direction of a relationship between 2 variables, you rely on correlation tests. Choosing the wrong tool means your entire analysis gets misaligned, so you definitely want to memorize this framework.
Step into the trachea with me for a second. Imagine we're in a BBC Earth documentary revealing the wet, organic texture and the living movement of our respiratory system. Here we have what we call the core trio. You've got the rigid, C-shaped cartilage providing structural support, the sweeping cilia of the epithelium moving mucus along, and the goblet cells secretly secreting that mucus in the background. When an examiner uses command words like compare or evaluate with these specific tissues, they fully expect you to identify both the similarities and the differences, and you have to support every single point with specific observable evidence from the stimulus they provide.
So, the absolutely crucial point here is how you handle unfamiliar contexts. When you are faced with experimental skills or totally novel data, you should rely on this 4-step sequence. First, deconstruct the given information and break it down into logical steps. Second, solve it systematically using really clear working.
Third, check for reasonableness. Basically, ask yourself, does this answer make biological sense? I mean, if your calculation suggests a human cell is the size of a tennis ball, you instantly know you've missed a step somewhere. And fourth, make justified recommendations. This sequence is your anchor when the exam throws a wild, unseen scenario at you.
And this brilliantly illustrates how we can apply this exact procedural framework to a really complex pathway. Let's consider the flow of electrons in cyclic photophosphorylation, which involves photosystem 1 and ATP synthesis. The absolute biggest pitfall right here is the generic answer trap. Students so often just provide a textbook definition of photoactivation or chemiosmosis that is entirely untailored to the specific data or graph provided in the question. You have to describe the exact flow of electrons exactly as it's depicted in the specific scenario you are given, not just dump what you memorized from the revision guide.
I really want to let this examiner quote sink in for a moment. The classic mistake is providing generic answers not specific to the question context. Always link every point to the given scenario. This right here is the dividing line between an average score and an A star. True mastery is transferability. Examiners are actively testing whether your biological principles can be applied, using evidence-based reasoning in entirely new, unseen situations.
Let's look at another highly dynamic system. Picture the cholinergic synapse. The action potential arrives at the bulb and boom, it triggers a massive high-stakes event. The sudden influx of calcium ions into the presynaptic membrane is the absolute critical trigger for neurotransmitter release. If you go on and describe this entire beautiful, complex process of vesicle fusion, but you fail to specifically mention the role of calcium ions, you completely lose the crucial process mark. Specificity is quite literally everything. Every single ion, every step, every term matters.
Let's move to and see how this builds when we zoom out to the digital landscape of bioinformatics. When you're comparing massive, data-heavy databases of nucleotide and amino acid sequences, what we call the context gap very often claims easy marks. A lack of unit precision, or just failing to define your key terms before you use them, creates a massive vulnerability in your answer. You absolutely must maintain a strict application to the specific biological context provided in that dataset.
Now for a quick Make It Stick challenge. I want you to take a second and predict. If you apply the wrong statistical test, say using a chi-squared test for comparing means instead of a t-test, on a really complex bioinformatics dataset, what exactly happens to your evaluative conclusion? Think about it for a second. Well, the answer is that your entire cause and effect chain completely collapses. Without the right test, your evaluation of limitations, alternative interpretations, and assumptions becomes scientifically invalid, and you lose all of those higher-order evaluative marks. So, what you should take away from this explainer is this. True mastery is understanding the why of biology, not just memorizing the what. When you can deconstruct the information, apply the correct statistical tools, and link your concepts to a specific context, the whole mystery of the examiner's mark scheme becomes brilliantly obvious. Apply this visual masterclass to your next set of past papers and just watch how your answers transform. But before you go, ask yourself this. If a single missing unit or one generic word can unravel a perfect memory, what other blind spots are hiding in your revision strategy right now? Keep investigating, keep questioning, and I'll see you in the next explainer.